Welcome back to the Deep Dive, where we take your source materials, the research, the notes, the highly technical papers, and turn them into actionable, memorable knowledge.
Today, we are strapping in for a journey into one of the human body's most complex and frankly hostile environments, the stomach.
It sounds a little counterintuitive, perhaps, but the stomach really is a silent internal battlefield.
It absolutely is. I mean, you have this incredibly strong acid, a pH, low enough to dissolve certain metals, and it's right up against this elaborate self-defensive layer of specialized tissue.
And when that defense fails, even just a little, it kicks off a chronic disease process.
Exactly.
So today we are diving deep into gastritis, and we're not talking about just a temporary upset stomach. We're talking about the long-term architectural destruction of this vital organ. Your source material provides a really forensic look at the entire process. We've got the pathophysiology, which details, exactly how the disease starts and progresses.
And the histopathology, which is how doctors actually see and confirm that damage under a microscope.
Our mission today is to understand the entire lifespan of this disease, from mapping the normal cell architecture all the way to tracing the sophisticated strategies of microbial evaders and even self-destructive immune responses. Okay, let's unpack this.
Well, gastritis, as your sources define it, is so much more than just inflammation. It's a sustained, complex interaction.
Right.
We're looking at these specialized bacterial agents, mainly Helicobacter pylori, engaging in what is essentially chemical warfare and biological deception against the host's immune system.
And that leads to this predictable tissue destruction.
Precisely. We'll explore those two primary drivers, H. pylori and autoimmunity, and then get into the standardized, really precise methods that pathologists use to map the resulting architectural disaster.
To really understand what goes wrong, I think we first need to appreciate what perfection looks like. We have to establish the architecture of the gastric mucosa, the wall itself.
That's the perfect place to start.
Because if we don't get the structure of these specialized cells, we can't truly grasp the gravity of their systematic destruction later on.
Absolutely. The stomach lining, or the mucosa, it's not uniform at all. It's actually neatly divided into two main functional territories.
And each is defined by the types of glands and cells they have, or they're doing completely different jobs.
Completely different jobs. First, you have what's called the oxyntic region.
Okay.
This covers the fundus, which is the top part of the stomach, and the corpus, the main body. You can think of this as the main chemical factory.
The production floor.
Exactly. Its glands are just densely packed with the cells that produce the actual digestive components.
So the primary workers in that factory would have to be the acid producers.
That's them. The most critical cell type here is the parietal cell, or you'll also hear it called the oxyntic cell.
And what do they look like?
They're physically large cells, often round or pyramidal, and they are highly acidophilic. That just means they take up acidic stains really brightly in a lab setting. Their entire job is acid secretion and, critically, the production of intrinsic factor, which is something we are definitely going to revisit later.
Okay, intrinsic factor. Got it. And what about the other vital cell type in that oxyntic region?
Then you have the chief cells, which are also known as zymogenic cells. They're a bit smaller than the parietal cells, and they exhibit basophilia, so they stain more of a bluish color.
And their job?
They're responsible for producing pepsinogen. That's the precursor to pepsin, which is what helps digest proteins. And then scattered among all these are the enterochromaffin cells, which sort of regulate motility and secretion.
So it's a very dense, very specialized area.
Extremely.
Yeah.
And that density and specialization makes the oxyntic region extremely vulnerable if the immune system ever breaches the defenses.
Okay, so that's the production core. Now, if we move away from the corpus and fundus and shift down to the mucus-secreting region, the antrum, we're looking at a totally different function.
Totally different.
It's more like a control center and a thick defensive shield.
That's a great analogy. The antrum, which includes the incisura angularis, is characterized by these deep surface indentations called gastric pits, or foveolae.
And this region is just packed with mucus-producing cells.
Abundantly so.
What are these surface cells actually made of?
Primarily foveolar cells. These are tall, columnar cells with a pale, eosinophilic cytoplasm. They have a very distinct look under the microscope compared to those dense oxyntic cells.
And they create that protective gel layer.
They create the protective gelatinous layer of mucus that physically shields all the epithelial cells beneath them from the stomach acid.
And I see here in the notes, deep within the pits, there are also mucus neck cells.
Yes, you can think of those as the reserve force. They are constantly maturing and migrating upward to replace the surface foveolar cells that are inevitably shed by the corrosive environment.
They're the sacrificial layer, really, ensuring the barrier stays intact.
They are.
This all sounds very neatly compartmentalized, yet your sources mention transitional zones or intermediate mucosa areas where these cell types start to merge. Why is identifying these boundaries so critical for a pathologist?
Well, pathologists need reference points. These transitional zones, the boundaries between the antrum and body, the body and cardia, the antrum and duodenum, they're where we see a gradual merging of the cell types. Damage often starts or is most pronounced right near these junctures.
So how does a pathologist definitively say, okay, I am looking at antrum tissue, as opposed to I'm looking at corpus or body tissue?
The visual contrast is actually quite striking once you know what to look for. The most informative indicator that you've moved from the acid-producing body into the regulatory antrum is the absence of chief cells.
They just disappear.
They disappear, and you see a radical shift in the gland structure. In the oxyntic body, the glands are long, simple tubular structures, densely packed, and the foveolae, the pits, are quite short.
Okay. And in the antrum?
Conversely, in the antrum, the glands become distinctly branched, and they're much shorter. In fact, approximately half of the antrum mucosal thickness is composed of those large mucus-producing glands.
So it's a total architectural shift.
A total shift. From long, straight enzyme-packed tubes to branched, mucus-heavy pits. That's the signal you're looking for.
So armed with this map of normal tissue, let's turn to the diagnostic confirmation of the disease itself. How is gastritis officially confirmed? What's the qualitative threshold that separates, normal low-level immune traffic from full-blown inflammation?
Right, because you can't just rely on the patient's symptoms. They can vary wildly. Histologically, we confirm active gastritis when we see a significant, non-negligible infiltration of inflammatory cells.
So you're looking for immune cells that shouldn't be there in those numbers.
Exactly. This means observing a marked increase in neutrophils, which is a sign of an acute active attack, or mononuclear cells, which is a sign of chronic ongoing inflammation across the biopsy sites. We're looking for clear evidence that the immune system is activated and engaged in a fight that it really shouldn't be having.
To ensure standardization and, maybe more importantly, to avoid missing patchy areas of disease, the source material really emphasizes a specific standardized mapping protocol, the Sidney system.
The Sidney system is absolutely fundamental. You simply can't diagnose gastritis reliably with a single random sample. It just doesn't work.
So what does it mandate?
The Sidney system mandates obtaining specimens from 5 distinct gastric locations. This includes 2 sites in the antrum, greater and lesser curvature, the incisura, which is that angular notch, and 2 sites in the corpus, again, greater and lesser curvature.
Why is this geographical mapping so crucial? I mean, if the stomach is inflamed, shouldn't one sample be enough?
Well, think of it like a viral outbreak in a city. You don't take a single sample from a random street corner and conclude the whole city is fine or the whole city is infected.
That's a good point.
Pathogens like H. pylori often don't colonize uniformly. They might settle really heavily in the antrum, leaving the corpus relatively untouched early on, or vice versa in some autoimmune processes.
So you need the full map to see the whole picture.
You need the map. And the other key mandate of the Sidney system is that those specimens must be placed in separate vials and carefully labeled by location.
That sounds like a simple administrative step, but it's clearly critical.
It's so critical. If a pathologist finds H. pylori in the antrum sample, but not the corpus, they can diagnose an antral predominant gastritis. If they mix the samples, they risk diluting the evidence of a localized patchy infection.
And you could get a false negative.
You could get a false negative or miss early signs of atrophy that only occur in one region. It enhances diagnostic fidelity exponentially.
Okay, with the architecture and the diagnostic rules established, let's introduce the primary enemy in this battlefield, H. pylori.
The master evader.
It's a flagellated gram-negative bacterium, usually transmitted through environmental contamination or fecal oral routes. Logically, it should be just instantly annihilated by the stomach's acid.
It should be.
But it's not, and this is the ultimate biological paradox. How on earth does this organism survive such a caustic environment?
Its survival is down to a single, incredibly elegant piece of biological engineering, the enzyme urease.
So it's not a passive organism?
Not at all. It actively modifies its immediate surroundings to create a habitable microclimate for itself.
Let's talk about the chemistry of that modification, because this is where the sophistication really begins.
So urease catalyzes the hydrolysis of urea, and urea is plentiful in the human body. When H. pylori breaks down that urea, it releases a key byproduct, ammonia.
And ammonia is, of course, a base.
Precisely. This ammonia immediately forms a dense chemical cloud, a protective layer, right around the bacterium.
It's like a personal force field.
It's like carrying a highly effective, localized, neutralizing smoke screen. Other bacteria get dissolved by the acid. H. pylori just changes the acid into a buffered solution, which allows it to move forward and colonize the epithelial surface.
So the path is chemically cleared. Now, how does it physically breach the final layer of defense, that thick mucus blanket?
Once it's buffered, the bacterium uses its flagellum, its tail for motility, which allows it to literally burrow through the mucus.
And it has enzymes for that too, right?
It does. It deploys mucolytic enzymes that help break down the thick, viscous mucus layer. This penetration lets it reach and firmly attach to the gastric epithelial cells. But critically, it doesn't typically invade the cells themselves. It adheres to the surface, right above the epithelial layer.
Here's where it gets really interesting. Because once it's attached, H. pylori begins its campaign of chronic destruction using this whole suite of sophisticated tools known as virulence factors. It's not just its physical presence, it's an active disruptor.
That's the crux of the pathology, really. These virulence factors serve multiple roles. They facilitate tighter cell adhesion, they cause direct cellular damage, they disrupt the critical tight junctions that keep the epithelial barrier sealed.
And most importantly, they engage in molecular mimicry and immune evasion.
Yes.
The sources specifically highlight CAGA, the cytotoxin-associated gene A. Why does this particular factor carry such long-term, ominous implications?
CAG is a toxin that H. pylori injects directly into the host's gastric epithelial cells. It uses a specialized structure, almost like a miniature syringe.
It injects it straight in.
Straight in. And once it's inside, GAGA just starts interfering with the host cell signaling pathways. It's a potent inducer of inflammation, yes, but its darker side is its strong association with oncogenesis, the development of gastric cancer.
So it's a major risk factor.
It's a distinct risk factor. The presence of CAGA-positive strains of H. pylori seems to drive chronically inflamed cells down a specifically destructive and potentially malignant pathway faster than CAGA-negative strains. It acts like a molecular switch for severe cellular dysregulation.
Let's pivot to the most incredible part of this strategy, immune evasion. The host body sees the bacterial antigens, it deploys its heavy artillery, the T cells and macrophages, but the infection persists, often for decades. Why do the T cells fail to clear this infection?
This is that inflammatory paradox we mentioned. When the body launches its targeted immune response, the T cells arrive, ready to kill the infected or damaged cells. However, H. pylori performs an extraordinary act of biological deception.
And what is that deception?
It forces the host's own gastric epithelial cells to express a molecule called B7H1, which you'll also see called programmed Death-1 Ligand 1, or PDL1.
PDL1. That is an incredibly potent molecule in immunology. You hear about it all the time in cancer research and immunotherapy.
It is. In a healthy scenario, when a T cell recognizes a foreign antigen, it seeks to kill the offending cell. But when that T cell encounters PDL1 on a target cell, it receives an inhibitory signal. It gets a message to stand down or do not engage.
So the bacterium is hijacking the cells.
It's hijacking the epithelial cells and turning them into biological white flags. It forces the epithelial cell to display this friendly fire avoidance signal, and by doing that, it paralyzes the very specific and potent T cell response designed to clear the infection.
The T cells just neutralize.
They're inhibited, they go quiescent, and the specific curative arm of the immune response is shut down.
That is truly astounding. The bacterium doesn't just survive, it uses the host's own cellular machinery to protect itself from the most sophisticated parts of the immune system.
Yes, and that allows the chronicity to really take root.
So the T cells are neutralized, but the infection is still causing damage.
Right, so the body keeps trying to fight it using nonspecific mechanisms. While it's inhibiting the T cells, the bacterium is actively sustaining this nonspecific destructive inflammation by inducing a massive increase in interleukin-8, or IL-8, production from those same epithelial cells.
And IL-8 acts like a chemical flare, signaling for immediate nonspecific backup.
It's the ultimate recruitment signal. IL-8 massively activates and recruits neutrophils, the frontline soldiers of inflammation, into the mucosa.
So you get this perpetual unresolved battle.
A perpetual battle. The T cells, which could solve the problem, are locked out. while this steady stream of neutrophils and other inflammatory cells constantly damages the host tissue, but can't eliminate the deeply embedded bacterium.
So the destruction isn't just caused by the bacterium itself, but by the host's own ineffective yet sustained inflammatory response. What's the eventual architectural cost of this endless, unwinnable chronic battle?
The relentless, non-specific inflammation eventually exhausts and destroys the very specialized cells that define the stomach's function.
They just get worn out.
They get worn out and killed off. Over time, we see the depletion of gastrin-producing G cells in the antrum, which regulate acid, and, critically, the wholesale destruction of the acid-producing parietal cells in the corpus.
And that depletion leads to severe long-term outcomes.
Two severe outcomes. First, atrophy, which means the thinning of the gastric mucosa. And second, the destructive progression toward intestinal metaplasia, where the body replaces the damaged specialized gastric tissue with something that looks like intestinal lining. The chronic fight fundamentally changes the organ itself. So if H. pylori demonstrated a masterclass in external invasion, then autoimmune gastritis, or AIG, represents a devastating internal catastrophe.
It's a failure of molecular self-recognition.
Exactly. The body's own immune system turns its highly destructive machinery against its most vital specialized cells.
This sounds like a more rapid, maybe more profound type of destruction because it's sustained by the body's own system. It's not reliant on a persistent external pathogen.
It is. It's T-cell mediated, and it is relentlessly self-destructive. Autoimmune metaplastic atrophic gastritis is driven by the T-cell mediated destruction of the oxyntic mucosa, so the corpus and fundus, and the creation of these self-attacking proteins called autoantibodies.
What are the primary targets of this devastating friendly fire?
The immune system specifically targets two irreplaceable components of that oxyntic region: the parietal cells, the acid producers we talked about.
And the intrinsic factor.
And the intrinsic factor.
Destruction of parietal cells, of course, means loss of acid, but intrinsic factor is critical for vitamin B12 absorption.
Correct. Intrinsic factor binds to dietary B12, allowing it to be absorbed in the small intestine. When the immune system destroys the cells that make intrinsic factor, B12 deficiency ensues, which eventually leads to pernicious anemia.
So in AIG, the body is simultaneously dismantling its chemical defense system and its vital nutrient absorption mechanism.
It's a two-front war against itself.
Given that H. pylori is the most common cause of inflammation, the source material explores a potential bridge between the two conditions. The crossover hypothesis. Does H. pylori ever accidentally start the autoimmune fire?
The crossover hypothesis is a critical piece of the puzzle. It proposes that in certain genetically susceptible patients who have pre-existing, long-standing H. pylori-induced atrophic gastritis, the immune response just gets confused.
How so?
Well, the T cells are trained to attack the bacterial antigens, but due to a concept called molecular mimicry, these bacterial antigens look incredibly similar to certain host antigens.
Specifically, components of the parietal cells and intrinsic factor.
Exactly. The immune system, having learned to fight the invader, essentially cross-references its target list and starts attacking the host's specialized cells because they look just enough like the bacterial threat.
So the H. pylori infection acts as the unintentional trigger that initiates this self-attacking response, even if the bacterium is later cleared.
That's the theory. It sets up the long-term, self-sustaining autoimmune damage that persists independently of the original infection.
Now, setting aside that crossover idea, there is also primary autoimmune gastritis, where the body just starts attacking itself, seemingly out of the blue.
In primary AG, the immune response targets the host antigen's parietal cells and intrinsic factor, irrespective of any detectable H. pylori infection. The body just launches the attack on its own.
And here we face a significant scientific unknown.
We really do. Despite extensive research, the specific molecular factors, the initial signals, or the precise genetic predispositions that truly drive the initiation of primary autoimmune gastritis, they remain unidentified.
It's a fundamental mystery.
It is. We know what the body attacks, but we don't know the exact mechanism that compels the immune system to break molecular tolerance in the first place.
Regardless of what starts it, crossover or primary, what is the eventual fate of the gastric tissue under this sustained T-cell mediated assault?
The hallmark of advanced AIG is the progressive replacement of the specialized oxyntic mucosa. Over time, the oxyntic glands are destroyed and replaced with aberrant cell types, leading to a profound architectural shift.
So the normal tissue is replaced with something else entirely.
We see the replacement of normal acid-secreting tissue with mucus cells and new abnormal gland structures. This replacement includes both the undesirable intestinal metaplasia and what's called pseudo-pyloric metaplasia.
And the destruction of the oxyntic region, where the acid factory is, means the body loses its most critical defense and digestive capacity.
Yes, creating a fundamentally altered and often pre-malignant microenvironment.
We've thoroughly mapped the sophisticated mechanisms of invasion and self-destruction. Now we have to shift to the visual, concrete evidence that pathologists rely on to confirm a definitive diagnosis from those biopsy samples.
The view from the microscope.
The first challenge is actually finding that tiny spiral-shaped bacterium, H. pylori.
Right. And the pathologist's starting tool is the standard stain used across all pathology, hematoxylin and eosin, or H&E.
How good is it?
H&E can detect the spiral-shaped bacteria, typically clinging to the epithelial surface, but its reliability is not absolute. We know it often misses subtle infections.
So, if H&E is insufficient, we have to turn to specialized staining methods to ensure diagnostic accuracy. Your sources highlight 3 key specialized methods. Let's start with the workhorse of the lab.
That would be the modified Jamsys stain. It's highly effective, it's simple to use, and it's cost-effective, which is why it's the preferred secondary method in many clinical labs. It dramatically improves visualization of the organisms against the background tissue.
So it just makes them easier to see.
Much easier. It ensures we don't confuse, say, mucus strands or cellular debris for the bacteria.
Then there's the more comprehensive technique, the Genta stain. What makes Genta a superior visualization tool?
The Genta stain is fascinating because it's a multi-component method. It combines silver staining, H&E, and Alcian blue.
So it's 3 stains in one.
Essentially. And the beauty of this combination is that it allows the pathologist to visualize 3 things simultaneously. The host tissue architecture from the H&E, the mucus layer from the Alcian blue, and the H. pylori organism itself from the silver. It paints a complete picture of the battleground.
That's incredible. Finally, we have the most strategically important stain for specific clinical scenarios, immunohistochemical, or IHC, staining. Why is IHC a must-have, particularly in patients who have been previously treated?
IHC staining is essential when you suspect the patient received treatment that suppressed but didn't entirely eradicate the infection.
Why does that matter?
Because when the bacterium is exposed to antibiotics, it can undergo a morphological change. It shifts from its spiral, rod-like shape into a round, atypical form called the cocoid form.
And that form is harder to see.
Incredibly difficult to detect with standard H&E or even Jamsys stains. IHC staining uses specific antibodies designed to bind to H. pylori components, irrespective of the bacterium shape.
So it will light up no matter what it looks like.
Exactly. This gives the pathologist the highest confidence, even if the organism is hiding in an atypical form, which prevents a misdiagnosis that could lead to a clinical recurrence.
Okay, let's move to the physical signs of damage. The histological features of H. pylori gastritis. What is the general rule?
The general rule is the presence of chronic inflammation, which typically presents as diffuse or nodular lymphocytic infiltration throughout the mucosa. The entire lining is just crowded with immune cells engaged in that long, unresolved fight.
How do we differentiate between, say, an acute flare-up of the infection and the long-term chronic damage?
In the acute phase, the visual clue is the presence of neutrophilic infiltration, a rush of those immediate, highly destructive inflammatory cells, often concentrating in the mucous neck region.
And you can see the damage they're doing.
Oh, yes. This active destruction is coupled with visible mucin loss and the sloughing or desquamation of the surface alveolar cells.
Yeah.
You can see the immediate chemical defense being actively breached.
And the chronic phase?
In the chronic phase, the visual hallmark is the development of lymphoid follicles. These are organized structures of immune cells that form when an infection has been sustained over a long period. Lymphoid follicles are highly abnormal in the gastric mucosa, and their presence is a definitive sign of long-term, sustained, organized chronic inflammation.
The sources also mention a condition called reactive gastritis, or gastropathy, which is defined by architectural changes driven by persistent low-grade irritation. This might not always be a massive H. pylori infection, right?
That's right. Reactive gastritis is often associated with factors like chemical reflux, bile acids, or persistent use of NSAIDs. It's a response to chronic chemical or mechanical stress, not necessarily an active infection.
So what does that look like histologically?
It involves a defensive architectural shift. We see marked foveolar hyperplasia, which means the surface pits proliferate and thicken dramatically to try and fortify that mucus layer. We also look for vascular ectasia, edema, and a thickening of the muscularis mucosae.
So these are structural reactive changes.
Exactly. The tissue is stressed, and it's rebuilding its nonspecific defenses, but usually without the heavy neutrophilic infiltration you see in an active H. pylori infection.
Now, shifting to the visual progression of the self-destruction, autoimmune gastritis, what defines the early microscopic changes?
Early AIG changes can be nonspecific, and frustratingly so. You might only see infiltrating lymphocytes and plasma cells within the lamina propria of the oxyntic mucosa.
So it's subtle.
Very subtle.
Right.
We might notice patchy destruction of the oxyntic glands, and sometimes the remaining parietal cells try to compensate, exhibiting what we call pseudo-hypertrophic changes. They look enlarged right before they're destroyed.
As the disease advances, what architectural breakdown occurs?
As the autoimmune destruction progresses, the inflammation becomes diffuse, a widespread lymphoplasmacytic infiltration. This is coupled with severe atrophy, so significant thinning and destruction of the specialized oxyntic glands.
And that's often masked, isn't it?
It is, often by a very prominent foveolar hyperplasia, where the surface layer tries desperately to compensate for the loss of the deep glands. And, of course, the inevitable appearance of metaplasia begins.
What is the pathognomonic or the absolutely defining feature that separates autoimmune destruction from H. pylori destruction, which can affect the whole stomach?
This is the critical fingerprint of AIG. The pathognomonic diagnosis requires the pathologist to observe those distinctive changes, destruction, and atrophy, specifically confined to the oxyntic mucosa, so the body and fundus.
And that has to be coupled with what?
This must be coupled with the crucial absence of inflammatory and atrophic changes in the antral mucosa.
So the antrum is spared.
The antrum is spared. Because the autoimmune targets are the parietal cells, which live in the body and fundus, the immune system generally leaves the antrum alone. H. pylori is typically a pangastritis. It affects both. If the antrum looks perfectly healthy while the corpus is completely destroyed, it is a definitive marker of autoimmune disease.
And finally, what does the end stage of this progressive destruction look like under the scope?
In the end stage, the specialized tissue is functionally gone. It's characterized by the complete lack of remaining oxyntic glands. The acid factory is shut down for good. We see maximal foveolar hyperplasia and often the development of hyperplastic and inflammatory polyps.
And paradoxically, the inflammation can go down.
It can. As the specialized cells are replaced by resilient metaplastic tissue, you often see a significant reduction in the inflammatory cells because the original targets of the autoimmune attack are simply no longer present. The battle fades once the enemy has been completely destroyed. This final stage, this transition to metaplasia, is perhaps the most serious consequence of chronic gastritis. Metaplasia means the body has given up on rebuilding its original specialized tissue.
It's permanently replaced it with a fundamentally different, often aberrant tissue type.
Yes, and that carries profound implications for long-term health risks.
The first type of replacement tissue we encounter is pseudopyloric metaplasia. This is also referred to as spasmolytic polypeptide-expressing metaplasia. This sounds like the stomach is trying to replace the specialized corpus tissue with something that just looks like antral mucosa?
That's exactly right. It's defined by the replacement of the high-functioning parietal and chief cells with mucus-secreting cells that mimic the appearance of normal antral mucosa.
And we think we know where these replacement cells come from.
A key insight here is that the precursors for these replacement cells are believed to be the chief cells of the original oxyntic glands, which suggests the destruction drives a reprogramming of the surviving workers.
But if it looks like antrum, how do pathologists distinguish the pseudopyloric replacement from the actual normal antral tissue we mapped out earlier?
The devil is in the details, specifically in the glandular architecture and the endocrine population. Pseudopyloric glands are coiled, they're less abundant, and they contain noticeably less mucin than true normal antral glands.
But the critical distinguishing feature is something else.
The critical feature is the absence of endocrine cells, particularly the gastrin-secreting G cells. Normal antrum is loaded with G cells for acid regulation. Pseudopyloric metaplasia has none. That absence is a definitive marker of the pathological change.
The second and far more concerning type of architectural change is intestinal metaplasia, IM, where specialized gastric mucosa is substituted with intestinal epithelium. Why is this considered such a severe warning sign?
This is significant because it's a fundamental shift away from gastric function towards intestinal function. The defining microscopic indicator of intestinal metaplasia is the presence of goblet cells.
Goblet cells.
If a pathologist sees goblet cells, which are mucus-secreting cells typically found in the intestine, they know the tissue has undergone an intestinal transformation.
And this IM can be subcategorized, correct?
Yes. We classify it as complete IM when the epithelium looks fully formed, resembling small intestinal tissue with clear brush borders. Or more concerningly, incomplete IM, which shows features more akin to colonic epithelium, often lacking those well-defined brush borders.
And within that incomplete category, we zero in on type 3 intestinal metaplasia, which is flagged as particularly high risk. What makes type 3 specifically worrisome?
Type 3 is the incomplete form characterized by goblet cells that specifically resemble colonic epithelium, and they contain distinct types of chemical markers called sulfamesins.
And pathologists track this one closely.
Very closely, because this particular metaplasia has been strongly and consistently associated with an elevated risk of gastric adenocarcinoma stomach cancer. It is considered a significant marker of precancerous change.
That sounds like a clear high-risk flag for the patient, but your source material includes an incredibly important caveat right after establishing that strong risk association. What is that crucial distinction?
The source material explicitly notes that despite the elevated risk association, type 3 is not considered an obligatory precancerous lesion.
That's a huge distinction. What does not obligatory mean in this context, and why is that detail so crucial for our listener to grasp?
It means that while the risk is undeniably higher if you have type 3 IM, the progression to cancer is not guaranteed. It is not an inevitable linear progression. Many individuals with type 3 IM will never develop cancer.
So it raises questions.
It raises profound questions about how clinically useful it is to focus so heavily on subcategorizing IM types in this highly detailed manner if the ultimate disease progression isn't a guaranteed consequence. It is a powerful risk marker, certainly, but it's not a predetermined sentence.
That level of nuance is exactly why we conduct these deep dives. Moving away from metaplasia, we should briefly acknowledge several other, less common but distinctive forms of gastritis identified histologically.
Absolutely. The stomach can react to unique challenges in some very unique ways.
Let's start with lymphocytic gastritis.
Lymphocytic gastritis is defined by an unusually high density of T cells infiltrating the epithelium. Specifically, the definition requires a minimum threshold of a massive influx of T cells, predominantly CD3+ and CD8+ lymphocytes, into the epithelial layer.
What does that suggest?
It suggests a sustained, intense T cell reaction, though the precise cause is often difficult to pin down. It is often associated with H. pylori infection and also, interestingly, with celiac disease.
And eosinophilic gastritis, which signals a major shift in the type of inflammatory cells present.
Eosinophilic gastritis is more of an allergic or hypersensitivity reaction, where the tissue is swamped by eosinophils, the cells often associated with fighting parasites and allergies. The diagnosis is confirmed by finding a significant pathological cluster of these specific cells in the tissue.
But you have to rule other things out first.
Critically, yes. The pathologist must rule out common associated causes, like H. pylori, parasitic infections, or Crohn's disease, before confirming this specific primary diagnosis.
Then there is this structural change seen in collagenous gastritis.
This is a rare condition characterized by the deposition of a thick band of collagen fibrous protein just beneath the surface epithelial layer, typically thicker than 10 micrometers. It's essentially a buildup of scar-like tissue accompanied by inflammatory cells in the lamina propria, restricting the movement and function of the epithelial cells.
And finally, a very rare form, emphysematous gastritis, which carries a shocking mortality risk.
Emphysematous gastritis is frighteningly rare, but also frighteningly lethal. It is characterized by the presence of air or gas bubbles within the gastric wall itself, usually caused by a severe gas-forming bacterial infection of the wall tissue.
And the prognosis is not good.
No. The source material underscores the gravity of this condition, noting mortality rates often climbing up to 60%. It's a sobering reminder that while most gastritis is a chronic endurance test, some forms represent an immediate life-threatening emergency.
This has been an extraordinarily detailed look at the microscopic warfare within the stomach. We started by mapping the normal architecture, understanding the fragile specialization of the acid producers and the mucus shields.
We then traced the sophisticated attack vectors of the microbial invader, H. pylori, a master of chemical defense and biological deception, and followed the self-destructive pathways of autoimmune gastritis. So, what does this all mean?
Well, I think the takeaway here is that gastritis is a progressive disease that fundamentally degrades the organ. The initial battle leads to permanent architectural changes, atrophy, the thinning and destruction of specialized glands, and metaplasia, the replacement of unique tissue with lower functioning substitutes.
It really highlights the extreme complexity and specialization of the body's defenses, doesn't it?
It does. For an external agent like H. pylori to succeed, it must use a suite of highly specific tools, uree, to neutralize the acid, Cago to induce malignancy pathways, and B7H1 to paralyze the T cells and enforce chronicity. That level of biological sophistication is why diagnosis is impossible without the precise mapping of the Sydney system and the targeted visualization provided by specialized histology. And it leaves us with the nuance we uncovered in that final section relating to risk assessment. We detailed type 3 intestinal metaplasia because it is strongly associated with an elevated gastric cancer risk.
Right.
Yet, the sources explicitly clarified that it is not an obligatory precancerous lesion. The risk is high, but the progression is not certain.
And this raises an important question for critical thinking that extends far beyond gastroenterology. If a condition like type 3 metaplasia, despite its serious correlation with cancer, is specifically noted as not leading to guaranteed progression, what other medical conditions are currently classified and managed with high anxiety, primarily based on correlation or association, where the definitive obligatory link to the worst outcome is scientifically missing?
That is something for you to mull over as you navigate risk and health information in the future.